Hello, everybody, and welcome to our spooky October version of the What's Up in the Watershed webinar. Today, we are going to be learning all things creepy and crawly under the surface of the lagoon from Dr. Holly Sweat of the Smithsonian. But first, a couple of housekeeping issues. You will notice that the chat feature has been disabled, but the question section is open for business. So this is a question and answer webinar, so please feel free to ask any questions you may have throughout the presentation for our speaker to answer at the end. Every question is a good question, and only me and the speaker can see them, so please don't hesitate to ask anything. She loves to answer questions. If you would like to watch this webinar again or share it with a friend, it will be available on our YouTube channel probably by the end of the day. You can find it through our website, onelagoon.org, or by going to youtube.com and searching for One Lagoon. If you have any ideas for what you'd like to see for our future webinars, please let us know in the questions section. We are always looking for the next hot topic to talk about. Now, without further ado, I would like to introduce our speaker for the day. Dr. Holly Sweat is a marine community ecologist with the Smithsonian Marine Station in Fort Pierce. That is by far the coolest title I've ever heard. Her work focuses broadly on biodiversity in benthic marine ecosystems and how microbes can change how marine communities are structured. So welcome, Holly.
Thank you, Jessie. I'm really excited to be here today. Whenever Jessie pitched this to me, the timing sounded great. I was super excited about like a Halloween themed sort of talk. So really happy to be here. And I guess I should probably start kind of at the beginning, a little bit about my background. First, I lead the Benthic ecology program at the Smithsonian Marine Station. And if you're unfamiliar with the term benthic, that just means bottom dwellers. So kind of everything that is on the bottom of the ocean or the lagoon, everything that grows on surfaces like dock pilings or boat hulls, all of that is kind of in our wheelhouse. And if you think about it, that's most of the life in the oceans. There's only a few things, some fish, some marine mammals that are not at all associated with the benthos. So we work on these bottom dwelling communities. And specifically, like Jessie said, we work a lot on the tiny stuff. I have always been really excited by all the tiny organisms in the ocean because we spend most of our time overlooking them, and yet they're super important. So if you kind of think about some of the other tiny organisms in our world, like the microbiome in your gut, if that microbiome is off, you're probably not having a very good day. And if the microbes in the estuary are off, the whole ecosystem is not having a very good time either. So a little bit about how I got sort of interested in all of this. Hopefully I can advance my slides here. There we go. So I'm a child of the 80s and the 90s, and Honey, I Shrunk the Kids was really popular at that time. And, you know, it's billed as a comedy, but if you really think about what your life would be like if you were shrunk down to an inch tall, it's terrifying. This whole world that you don't think of suddenly becomes a little bit more threatening. So something like a bowl of Cheerios could end up being a death trap. And it also affects the way that we would interact with the world around us, like an ant in the backyard. And so that idea just sort of permeated my career. I started thinking about what would it be like if I was under this microscope with these animals? What would my world look like? So that's kind of what drives all of my work. And one of the things that we focus on mostly when we are focusing on the work in my lab is how it relates to restoration and conservation. And one of the big projects that we're involved in is something called the Comprehensive Everglades Restoration Plan, or CERP. So many of you probably have heard of this. You've heard of Lake Okeechobee releases. You've heard of issues with Florida's hydrology. For those of you who haven't, I'll just give you like a brief history of all of it. Historically, the fresh water in the Florida Peninsula originated north of Orlando in what's called the Kissimmee chain of lakes. And it would flow south through Lake Okeechobee, through the Everglades National Park area, and out into Florida Bay. And then in the early to mid 1900s, there was a big development boom in Florida, of course, and there was a need to kind of literally drain the swamp. And so a massive system of canals that was added onto over the years was constructed. And that shunts most of the fresh water out to the Gulf of Mexico through the Caloosahatchee River and at Fort Myers, and then out to the IRL and the Atlantic Ocean through the St. Lucie estuary, just south of where we are in Stewart. So this creates a lot of problems. Obviously, fresh water in a marine environment can be problematic for those animals, but that fresh water is also carrying a lot of other things. It has nutrients, sometimes harmful algae. It has suspended sediments that can rain down and blanket the bottom of the estuary and smother things. So about 20 years ago now, a big project called SERP got started to sort of return a more natural hydrology to Florida's peninsula. And that's an adaptive management scheme. And so the Army Corps of Engineers, the water management districts, a lot of folks are relying on experts that monitor all different parts of the system to give them good advice about how restoration is going and what the next steps are that we should be taking. So there are people that are looking at fishes, seagrasses, alligators and crocodiles. And in my lab, we look at what are called infaunal invertebrates, or infauna. So these infauna, you've probably seen a lot of them before. These are tiny invertebrates that live in the sand and the mud in our estuary. Most of them are less than half an inch long. They include things like worms and snails, shrimp, brittle stars, and they are world-renowned as great indicators of environmental health. And the reason that they're used so often is for a couple of different reasons. First off, they are literally stuck in the mud. They can't go anywhere. So whenever there's a disturbance, they can't swim away like a fish can. They also reproduce pretty quickly, so they better mirror the environment around them than something that has a longer lifespan. They're also super abundant. So just to give you an idea, based on our own data, our 20 years of monitoring data, of how abundant they are, if you are the average adult shoe size in the US, which, fun fact, is a women's 10.5, which is equal to a men's 9. If you are that shoe size, you put your foot down on the estuary floor, you have a good chance of stepping on nearly 9,000 of these animals just within the area of your footprint. So I want you to take that in for a minute and kind of hang on to that idea of scale. 9,000 of these animals can be living within the area of the bottom of your foot. So these are the reasons that we monitor infauna. And we've been doing that for, it'll be 20 years next year. We do that at 15 sites running from around the Harbor Branch area, just north of Fort Pierce Inlet, and then all throughout the St. Lucie estuary. And I wanted to kind of bring you along on one of our collecting trips and show you how we collect these different animals. So this is a video of what's called a ponar grab. This is the sediment grab that we use to get basically a big handful of sand that has all of these animals in it. So we lower it over the side of the boat. It has a pin that holds these claws open, kind of like the claw game in an arcade, if you've ever played that. Then when it hits the bottom, that pin releases, it grabs a bunch of sand. We pull it back up. Video's lagging a little bit. I have a lot of videos in this presentation. And then we transfer it over to a sieve. So the entire sand or mud sample gets dumped out into this sieve. And then we have to kind of sift through it, kind of like you're panning for gold. And that gets a lot of that fine-grained sediment out and retains the animals that we are interested in looking at. So it kind of does a pre-clean on the sample. And then what we have left in the sieve gets preserved, goes into a jar, and then we take that back to the lab for processing. So of course, we're looking at this benthic community, and this is an example of our samples here. If you're curious, the reason that they are pink is because we use a dye called rose bengal, which dyes all organic tissue, including your fingertips if you get it on your hands, and it allows us to more easily pick the animals out of the sediment. We also look at any other big animals that we happen to notice when we're out in the field, things like big sea stars, any seagrass if we find it. We measure different characteristics of the sediment, like organic matter and grain size, and we also look at water quality and weather. So when we get these samples back to the lab, the real work really begins. We have weeks to months of processing. One day in the field can be equal to a few months in the lab. So to date, we've actually looked at nearly 3,500 sediment grabs since we started this monitoring program. We have a list of about 1,100 different species that live in these sediments. And by now, we've counted well over 1.5 million individual organisms. And all of that work goes into creating assessments and reports for the Army Corps, for the Water Management District, for Congress. That gives us an idea of the health of the ecosystem. So this is one from very recently, just showing the letter grades basically that we get for different sites. So you'll see sites in the Indian River Lagoon tend to do better. And then as you go up into the St. Lucie estuary, closer to Lake Okeechobee and some of those other areas of freshwater outflow, the grades go down. And we are relying on these little monsters, these little invertebrates, to generate these grades and to tell stories about the ecosystems. And so I thought what would be fun today is just to kind of give you a few examples of those animals that we see every day. So if you are a horror movie buff or just a pop culture buff, you might notice some of the references I have on these next few slides. So the first couple of things that I wanted to point out are the animals that we see that have tentacles or that appear to have tentacles. And the first couple are what we call spaghetti worms, and another type of polychaete or worm called mud owls. So this is a spaghetti worm, and it's kind of all balled up in the dish here. But if you can see, this is the tail end. Hopefully you can see my cursor. And it kind of coils around like this. And then the head end has all of these tentacles. Now, in the wild, this worm would be burrowed into the sand so that the body is below the sand, and then the tentacles are sticking out on top of the sand, so kind of vertically. And those tentacles are extremely stretchy. So if you have a worm that's, maybe 1/2 inch long, burrowed into the sediment, those tentacles can extend out inches from where that worm is. They're really sticky and they grab a hold of all sorts of stuff on the top of the sand, mostly detritus, bits of dead, decaying matter. They grab it like flypaper, they reel it in, and they eat it. And then my next example is a little bit different. This is also a polychaete, so it's a segmented worm, but where you think it might have tentacles, these are actually gills. So again, it burrows into the surface sediments and it extends these gills up to the top of the sediment so that it can breathe more easily. And it also feeds on organic matter in the sediment. We call these deposit feeders. But I wanted to wait to play it because the movement is very different. So this is how they move through the sediment. You might expect the gill part to be the head. It's not. That's the tail end. So it's actually burrowing into the sediment head first. And this sort of motion where it's extruding its head and then sucking it back in allows it, of course, to move around, but it also allows it to grab big mouthfuls of sediment, pull it in, and then it extracts the nutrient-rich part, all the organic matter, and it excretes clean sand. It's a little bit like a filter or a vacuum cleaner. So both of these little monsters are examples of animals that do a really good job at cleaning surface sediments. They're mostly just in the top couple of centimeters or quarter of an inch of the bottom of the lagoon. The next example I'm going to show you is something that penetrates a little bit deeper. A lot of these worms will go several inches into the sediment. This is one example. This is called Litoscoloplos, and it's a type of polychaete. It looks huge in this picture, I know, but the maximum length for these is only about 3 inches. So still, I guess in our world, it is pretty large. These are deep burrowers, and they engage in a really interesting feeding strategy that a lot of polychaetes have. So it'll cruise along the top of the sand, and it feeds a lot on what are called biofilms. So these are sort of algae that encrust the surface of the sand, things like diatoms and other microalgae. It's going to feed on these films. It'll dig down through the oxic layer, which is oxygen rich, and into the area of the sediment that's deep down and has no oxygen, what we call the anoxic layer. It will defecate out that biofilm down in the anoxic layer, then eat some more stuff while it's down there, climb back up, and start the process all over again. So in this way, it's doing a really good job of cycling nutrients and creating more oxygen deeper down in the sediments. It's also super tolerant to disturbance, so it lives in areas that are pretty polluted. Like I said, it helps clean the sand, and it's also a major food for small fishes like killifish. Next up is skeleton shrimp. I don't think I could talk in a sort of Halloween themed talk without mentioning skeleton shrimp. And I call them our little aliens. These are from the family Caprellidae. And fun fact, they are very closely related to another, caprellids are, I should back up a little bit. Caprellids are amphipods, so they're related to shrimp. They're not true shrimp. And they're very closely related to another amphipod that is thought to be the inspiration for the first alien in the Alien movies. And you can kind of see a little bit of a similarity there with the silhouette, but I'll show you some videos of it as well. So if you keep your eye right here, over here we've got a brittle star, but I want you to look right here on the screen as I play the video. This is a skeleton shrimp or a caprellid, and this is actually a female. So you can tell the females from the males because they don't have big claws and they carry the eggs on their abdomen. So you can kind of see when I play again, she's fanning her abdomen back and forth, and that's to keep those eggs oxygenated. And then just in a second, you'll see the male. Here comes the male. So you can see he's got really big claws, doesn't carry any eggs. Again, these are only about 1/2 inch long. And here is an image of the legs on these skeleton shrimp. So they have these modified appendages that are like hooks that allow them to grab onto seagrass blades or algae. And they spend their whole day kind of grabbing a hold and doing this bowing motion in the water to catch different things floating around in the water column and helping to clean up the water. These hooked legs are so strong and so precise that if you're holding one in your hand, they can grab a hold of the ridges in your fingerprint. And it's pretty hard to get them to let loose. So Pretty tenacious little things. Okay. And then lastly, I feel like I sped through this, but I've probably been talking for a little while. Lastly, I want to talk about sea spiders. Now, these are not true spiders. The spiders on land that we maybe sometimes are afraid to interact with are actually in the class Arachnida, and sea spiders are in the class Pycnogonida, but they look very similar to your house spiders. Here's a video of one. This is a good segue because this is a video of one kind of having a little bit of a fight with a skeleton shrimp. So this sea spider is probably less than the size of a dime. It's, again, very small. Sea spiders are known for kind of being like Velcro. They end up having a lot of stuff stuck to them because they have these little hairs, and they're pretty slow and can't clean stuff off. So when something like this skeleton shrimp attaches to it, it doesn't have the ability to fight it off very well. They're also interesting because they all of their organs are in their legs. So they actually breathe through their legs. And there's still a lot that we need to learn about them, but they move really slowly, and they're probably thought to be part of the ocean's cleanup crew because they will amble along, and I'll show you a video in a second, slowly on the benthos, and they will feed on soft-bodied material. So that probably includes both live and dead organisms. And like I said, this one's very small. We only get small ones in the IRL, but I wanted to show you a picture of a larger one for scale. This is my postdoctoral fellow, Dr. Jessica Zempfenig, and she actually did her dissertation work on deep sea Antarctic sea spiders. And this is one of the common ones that they pulled up on their cruises that she has just on the top of her head there for scale. So they do get very large. This one can get to be, I think, up to 1/2 meter in diameter. And to show you what it looks like in its native habitat, this is a video of one, again, ambling along very slowly, and it looks like it's feeding on some sort of organism there. So these are hardly a threat to us. Again, they live in very deep water. They move really slowly. So they kind of be like running from those old school zombies that can't run very fast. And then I wanted to keep this up to show you a picture of one that's sort of swimming along. They're pretty neat when they swim like this. And advance my slide here. Doesn't want to go. Oops. There we go. So again, I feel like I just breezed through this whole thing, but I wanted to end by really thanking everybody in my lab for their hard work. This benthic monitoring that we do is something that can't be done alone or even by just, a couple of people. It really takes a team effort. And there are several people that I want to thank that have been involved in this over the last couple of years. And then if I put all the past members of the Benthic Ecology Lab on here, I don't think I'd have room for anything else on the slide. It takes really a village to do this kind of work. I also want to thank all of our sponsors that have, monetarily sponsored this work or have given us other resources over the years. So with that, I am happy to take any questions you might have.
Awesome. Thank you so much, Holly. That was great. Let me turn my camera back on. So to me, I kind of feel like the mud owls might be the Roombas of the ocean floor,
Oh, that's nice. I like
right?
that.
Because they're like
I'll give you
cleaning
credit when I
everything
use that in the future.
up. And then I don't know how I feel about putting sea spiders on my head. I don't necessarily like normal spiders. I respect them enough, but I don't think I would put it on my head. So your lab is very brave. Okay, so we do have some questions from the chat. We're going to start off with the first one and a really fun one. What is your favorite micro critter?
Oh my gosh. I don't know if I have a favorite.
After
I
this,
always,
I think mine's the mud owl. I really like that.
yeah, well, I was gonna say that I always go to that video when I show people how cool these animals are. So it's probably in my top. I also really like skeleton shrimp because
Oh.
I like the way they move. I find them to be like, very polite. They're always bowing. So, yeah.
Kind of like praying mantis almost. That's kind of what I thought of when I saw them.
Yes, the claws especially are very mantis-like.
So, for any of these animals that, or their cousins that may live in the lagoon, do you know if any of them feed on the epiphytes that are on seagrass? And epiphytes
Yeah,
are little algae and stuff that hangs out on seagrass, little plants that hang out on plants.
yeah, that's a really good question. There's a bunch that um, will there's a lot of animals that are exclusive to the sediment, so they'll never crawl up on a seagrass blade. There's some that are exclusive to the grass blades, and then there's others that just kind of go back and forth. And the ones that do feed on seagrasses, there's a lot of the amphipods like skeleton shrimp and other closely related species that'll sort of inchworm along the grass blade, and they'll pick off things like hydroids, little bits of algae, diatoms, all sorts of stuff that grows on those grass blades. There are also a lot of snails that will crawl along and just they're a little bit like a Roomba actually, because they'll sort of scrape the biofilms, the algae off of the grass blades in this circular motion, and sometimes you
Yeah.
can actually see their track.
Oh, that's cool. Next time I do seagrass surveys, I'm gonna have to look closer and see if I see any skeleton shrimp,
Yeah.
but hopefully that would be neat. So another question we have is, are you seeing any increasing or decreasing trends in abundance with your research?
Mm, yeah, that's a good question. Yes and yes. It kind of depends on where we're looking. We are, I guess, fortunate that we monitor across this wide environmental gradient. So we have some sites that are doing much better than they were maybe a year ago. We're starting to see seagrasses come back, and so we expect that we're going to see a whole lot more diversity and abundance. And then we have some areas that are really disturbed, areas that are close to those Lake Okeechobee releases and other releases from inland, and those unfortunately have been declining over the years.
Gotcha. Okay. If you could be any microbe creature, which one would you be and why? They also say that you can create your own creature if you'd like.
Oh, wow.
I know that's a hard one. I feel like,
I feel like I'm going to have to sleep on this one.
right?
I really, I tend to gravitate toward the crustaceans, I think. I
I could see
really
that.
like amphipods. I like stuff that sort of skitters around at the surface. I'm a little claustrophobic, so I can't get behind, digging deep into the sand.
Okay, I
Um,
like that.
so yeah, something like that.
I feel like I would dig down because I'm very much a Netflix and chill type gal. So
Oh
if I
yeah.
just like dig down and have the food come to me and just vibe, I think that's that's what I would like to do if I were a
Just
micro.
get away from it all.
There you go, exactly. Can you explain, bringing it back to seagrass, can you explain the direct link between benthic animal health and seagrass health? And is
Oh,
that
yeah, that's a good question. We are actually looking into that right now. I have a new collaborative project with some folks that is looking at in fauna, these little invertebrates in seagrass beds. And some of the work has been done already, but we still don't know a lot about the details of that link. So we do know that grass beds tend to harbor more in fauna, and it's probably sort of a give and take relationship there. So in fauna can help stabilize the sediment. They create these tubes and other structures that can keep the sand from blowing away when we have a storm. And that, of course, is going to help seagrasses stay in place.
Hmm.
They also, like I mentioned, oxygenate the sediments. So chemically, they're more conducive for healthy seagrass. And then in turn, the seagrass is going to create some refuge for those invertebrates, some sort of structure for them to build their homes around.
Gotcha. Okay.
Yeah, so we're still trying to tease apart some details like what infaunal species are there that are out there that could be potentially used for seagrass restoration? Or if you're going out and scouting a place to figure out if it's suitable to plant seagrass, are there key species that exist out there that can tell you that's a good spot?
Yeah, so I think that really ties into our next question of are the abundances different between seagrass areas or submerged aquatic vegetation areas and non-submerged aquatic vegetation areas?
Yes. Yeah, when you have an area that has seagrass, you can expect, and an area adjacent to it with no seagrass, you could expect to have probably orders of magnitude greater diversity and abundance of fauna in the grass bed.
Very cool. Okay. I think this question is a little bit broad, and we may not be able to answer it using our benthic creatures, but um, are we seeing the health of the lagoon increasing or decreasing? I know up by us in the northern region, we are seeing a resurgence of seagrass. Water clarity is coming through. Everything's looking great, but that may not be the case down south. So, what are you seeing currently?
Mm-hmm. Yeah, that's a tough one. And I think, again, I have to mention it just kind of depends on where you are in the
Yeah, right.
estuary. We have a huge estuary. I mean, as many of you know, it takes up almost a third of Florida's east coastline. So, um, what's true in one area is not going to be true in another. And I'm really happy that we were out this summer and saw the seagrass coming back. And I just crossed my fingers that we stay in this upward trajectory. So I'm feeling really hopeful because of that. But there are still areas that are heavily impacted. And anytime we get a hurricane come through, like we just had, there's going to be a lot of fresh water carrying a lot of junk that we don't want. And that's going to get dumped into our estuary and create problems.
Yeah, so hopefully we'll continue seeing that upward trend. And if you have any more questions on that, please feel free to reach out to either of us via email. We'd love to chat with you about it. So 19 years worth of monitoring, that's a really long time. What was the impetus for starting this at the beginning?
Yeah, so this all got started kind of back at the beginning of when this SERP project began. So most of the monitoring for the Comprehensive Everglades Restoration Plan started in 2005.
Mhm.
And whenever they were forming the monitoring, they knew that they wanted it to be adaptive and they wanted to get a good holistic picture of what's going on in the estuary and how it's changing over time so they can have a good baseline and then know how the things that they implement like water retention areas or other projects to try to clean up um, the peninsula and try to reroute water if those can be successful, if those are being successful. So I think I might have gone down a rabbit hole a little bit on that one. I don't know if
No,
I answered your
it
question.
started because of SERP, and I think that's awesome. I
Yeah.
mean, that's a great reason to start, and I can't believe that 2005 was 19 years ago, but here we are.
I
Um,
know.
so
I know.
isn't that crazy? I don't know how much you are involved in muck removal. I know that it's happening down in the central and southern parts of the lagoon, but is there any data comparing populations in the area before muck removal and after muck removal?
Yeah, that's a good question. My group is not directly involved in muck removal, but we do have monitoring going on in areas that I know that muck is planning to be removed. I think the North Fork of the St. Lucie Estuary, they're planning a muck removal project. I think Florida Institute of Technology has had some before and after muck removal infaunal studies up in
Okay.
the Turkey Creek area in Melbourne.
So that's with Dr. Austin Fox's lab, right?
I think it was initially with Kevin Johnson's lab. I
Okay.
think Dr. Fox might have taken it over. Yeah,
Gotcha. So if you're doing the data now and there's a plan for muck removal, there's a potential that you'll have after data, but it may take a couple of years.
right. Yeah,
Oh,
it's going to
okay,
take a
yeah.
little bit of time for stuff to move in after we take all the muck out.
Awesome, awesome. How would you say that, and I feel like you did touch on this a little bit, but how do benthic organisms contribute to the overall health of the Indian River Lagoon ecosystem?
Mm. Yeah, so they do a great job of pulling nutrients out of the sediments and kind of, I think I alluded to them as vacuum cleaners. So
Yeah.
they're sucking up a lot of the nutrients that are on the bottom. Other ones like clams are really effective water filterers. So they're cleaning the water column. And at the same time, they're creating structure and stabilizing the sediments, like I mentioned, for seagrasses. So they're actually creating sort of the base of this ecosystem, the seagrass habitat that then allows it to flourish. And they're a super important food source for almost everything that feeds on the bottom of the ocean.
Gotcha. So really they are like a keystone community for the lagoon.
Yes.
Like the lagoon would not be able to survive without having these populations of microbes.
Yeah. If you got rid of them, it would be a desert for sure.
Okay. Are there any emerging technologies or methodologies being used to monitor or study the microbial communities?
Great question. Yes, there is. One of those things is environmental DNA or eDNA, and that's something that we've started employing this past year, or this year actually, to help monitor our sites. So environmental DNA is basically the um, bits and pieces of animals, like bits of mucus, scales from fish,
Hmm.
all of that stuff that they leave behind, kind of like when we walk into a room, we might shed hair or, bits of skin cells. That's our environmental DNA. This is happening in the ocean all the time.
Hmm.
And so we can take advantage of that by taking a sediment sample or taking a water sample and sequencing the DNA from it. And then we know what has been there recently. So it's a really great way to monitor an ecosystem without um, having such a direct impact. We don't have to collect as many animals, and the time component it's far shorter. It's a lot easier to sequence DNA than it is to sit behind a microscope for months and pick all of
Right.
these animals apart and ID them. So there's that, and then there's also AI technologies that are being developed
Okay.
um, using artificial intelligence to look at pictures of these animals and identify them. And then once you get the AI trained up, you can go through pictures really rapidly and get a sense of the community faster than a technician could in the lab.
That's cool. I like that. But then, I mean, will AI be able to put sea spiders on their head? I don't think so.
No, it's they're
There's
definitely going to have
trade-offs.
limitations. Yeah. Yeah.
How would you say the biodiversity of IRL benthic creatures compares to other estuaries?
That is a great question. And that is something that we don't really have an answer to yet.
Okay.
So I run the IRL species inventory, which aims to catalog all the life we have in the watershed. Unfortunately, there are not similar programs for a lot of estuaries in the country or around the world. So, we definitely are a biodiversity hotspot, but we don't know quantitatively how we compare to other estuaries.
Gotcha. Okay. And I did share that link to the IRL species inventory in the chat for anybody who would like to explore it. It is pretty fun to check out and see some of your favorite creatures in the lagoon. So one last question before we let you go today. How do you feel that human activity such as pollution or boat traffic impacts benthic organisms and in the lagoon?
Well, pollution definitely does. Anything that we dump into the water column that makes it all the way down to the bottom of the water is going to impact them. So when we have, for example, a big nutrient release, say there's sewage in the water and it causes an algal bloom or something like that, that's going to impact the critters that live in the sediments lots of different ways. One of the things could be if we have an algal bloom that starts because of excess nutrients, that's going to suck up oxygen and it could suffocate these animals. But there's a lot of different examples like that from different disturbances that we create.
Awesome. Thank you.
Yeah.
All right, so Halloween is next Thursday. If you guys dress up as a spaghetti worm, as a mud owl, as a sea spider, we want to see it. So please feel free to share your spooky benthic creature Halloween costumes on our Instagram or our Facebook page at One Lagoon. And thank you so much for joining us today, Holly. We really appreciate it.
You're welcome. I had a great time.
Thanks, everybody. Have a good day.
Bye.